Bon, on va commencer. Je vais commencer en français, puis je vais parler en anglais après.
Non, non, non.
Ah ouais? Non, aujourd'hui, on a la chance d'avoir Mattia Saccò, donc je vais vous le présenter dans quelques minutes. Donc, on essaie de faire un séminaire par mois. Donc, je ne sais pas si on arrivera à en faire un pour le mois prochain, parce qu'on est assez pris au niveau du labo, mais donc on essaie de maintenir un format de un par mois avec parfois des séminaires fermés qui ne sont pas ouverts à tout le muséum. Mais là, l'essentiel, c'est qu'évidemment, toutes les personnes de Boréa et extérieures à Boréa sont invitées à assister à nos séminaires. Donc, je vais essayer de parler en anglais. Today we have Mattia Saccò, who came from Australia, but is in Italian, in fact. He came from the lab at Curtin University in Perth. Mattia will be a guest researcher at Borea from January this year to 2026, so in December. And he's a member of a trace on environmental DNA labs at Curtin and a leader of subterranean research on groundwater ecology. And then I will give you the yes, to have the camera in front of you.
Okay, great. Thank you very much. And thank you everyone for being so kind to me during the last 2 months, despite I lost the key and badge after 3 weeks. Right? But I found them. I found them the other day. And so today the goal is to showcase a bit of a research that we are doing at Trend, which is the group I belong to at Curtin University in Perth, and also to show you some of the research more specifically on groundwater ecology and hypersaline ecology, using different techniques, including eDNA, stable isotopes, and biogeochemical tools more in general. Very little about me. I studied in Italy. I'm from Italy. I'm from Parma, the capital of cheese. Ooh, that's controversial to say in Paris, right? I have a bachelor's degree in ecological biology and then another bachelor degree, weirdly enough, from Spain on the same topic because Spain wouldn't recognize my degree in Italy. So European Union there wasn't really present. I have a master's degree in biodiversity, and then I completed my PhD in 2020 at Curtin on the functional ecology of a subterranean ecosystem. I'm a lecturer at Curtin University. And as Nicolas said, I'll be here until December 2026. So more or less 2 years as a guest researcher at Borea. Thank you guys for giving me this opportunity as well. As I said before, the goal of today's speech is to show you a bit of the research that we do at Curtin University with the main objective to foster inter and intra-group collaborations. So I'll be talking about Trend Lab and Search Group. Research projects, as I said, mainly focusing on groundwater ecology and hypersaline biomonitoring, and near future endeavors, including ecological modeling and testing novel biometric tools such as eRNA. Trend, pretty sure that you never heard about Trend, stands for Trace and Environmental DNA Lab. At Trend, we do not only work on eDNA, there's also a big chunk of the research done on genomics, which will refer to what happened on the way in between eDNA, which is the present, and ancient DNA, which is the ancient DNA in the environment. It turns out that once you search for eDNA research on Scopus, Trend specifically, and Curtin University in this case, that's what we are showing there, is the leading institution in environmental DNA research in terms of publications. It's a fairly, in terms of numbers of people, it's fairly decent, smaller than Borea. We have a leader, Professor Morten Allentoft, and 30 researchers belonging to the research branch called Trend, and there's also a more business branch called eDNA Frontiers. Within Trend, there's definitely similar to Borea, different research groups. There's the one that I lead on subterranean ecology. There's another group on human microbiome. There's a group on ecological restoration in mine sites. There's a big group on marine biology and coral research as well. Search group members, very quickly, myself, my research focuses mainly on functional ecology and multidisciplinary approaches. The co-leader is Nicole White, 25 years experience on molecular ecology. There's Mieke van der Heijden, who's really good at trying to use eDNA as a tool to improve biomonitoring in groundwater ecosystems. And then we have a researcher specialized in phylogeny and species identification. I'm currently supervising 2 PhD students. The projects focus on ecotoxicological modeling and on the, again, modeling, but more on the statistical side of things. 3 quick definitions. Stygofauna, pretty sure that you probably never heard about that. That's exactly the way I reacted when I heard it for the first time. Stygo what? It pretty much refers to the invertebrates of groundwater ecosystems. So subterranean aquatic reservoirs hosting invertebrate biota. Calcrete, another word that if you're not a geologist or into groundwater ecology, you probably never heard. It refers to secondary sedimentary deposits formed by the precipitation of calcium carbonate, similarly to karst, from shallow groundwaters along the path of a paleo drainage channel, and also hypersaline. I'm pretty sure that this one is perhaps more familiar. It refers to water bodies where the concentration of salt exceeds 35 grams per liter, which is the salinity conventionally of the oceans. Great examples over there. You have the Great Salt Lake, pretty sure that you heard about, the Dead Sea, the Lake in Australia, and the Salar Atacama in Chile. One last definition, eDNA, not sure if you're familiar with this tool, is getting more and more popular for a series of reasons as a biomonitoring tool. It refers to the total pool of DNA isolated from environmental samples. So the definition is very broad. It's basically genetic material derived from biotic activities. So any single biota, any single organism in an environment would shed DNA. If we target that DNA, we are able to detect that. It's pretty much forensics applied to ecology or environmental conservation. There's 2 non-destructive categories for eDNA. There's organismal. Of course, with microbes, we cannot talk about tissue. We talk about the whole organism, and extra-organismal in terms of, for example, the feces or the shed tissue from a fish. You have there on the right the 4 stages classically of the eDNA. You collect the samples, then you extract the DNA from the environmental sample, which could be sediment or water. You amplify target DNA barcodes, and then there's a sequencing, which is the most conventional part of this DNA barcodes. So you're pretty much assigning the certain barcodes that you can find within the environmental sample to a database. Is applied in a plethora of aquatic ecosystems for studying assemblages, food web interactions, invasive species. You usually have 2 approaches. One is the needle approach, so you target one specific species that could be endangered or invasive, or many other categories. Or there's also the high-stack approach. You go after the community. You collect a sample from a lake and you want to see what's the community in terms of invertebrate diversity. The field is booming, as you probably heard or you noticed during the last few years. I would just wanted to give you an idea. In 2 months, if you type groundwater ecosystems, don't know how much you work with groundwater ecosystems. They're interconnected to surface ecosystems most of the time. They provide hugely important services, drinking water provision. If you think about cities of like Vienna, Oslo, depending entirely for the potable water on groundwater ecosystems. They're increasingly used for industry, for cooling down. For example, we have a supercomputer at Perth University and it uses the water from the groundwater to cool down the machinery within this facility. But it's also important from an ecological point of view. You have a mostly overlooked diversity over there in terms of vertebrate and invertebrate species. It's important in terms of conservation of groundwater dependent ecosystems, and it's also crucial for the microbial activity that the resident biota is exerting in maintaining the water quality. Here, I'm assuming that if I do this, okay, I can do this. Okay, this is collaborative paper that we published relatively recently, and we found out through some relatively complex modeling that groundwater is actually a global keystone ecosystem. That means that preserving groundwater is actually essential for preserving surface biodiversity. The numbers that we found were quite interesting. I reckon if you think that over 50% of global areas around the world have medium to high groundwater surface water interaction, and this has an impact on the modeled biodiversity that we could assess. If again you think that 50% of global areas with high surface biodiversity have some sort of groundwater interaction. This is a field of research that is still massively underexplored, but there's more and more evidence on the importance of groundwaters. There's, of course, biota is not just a Tupperware full of water, and this biota is essential because, for example, stegofauna is important for burrowing and bioturbating the sediment. That way the oxygen is, the water is more rich in oxygen and the ecosystem is richer. They purify water from excess of contaminants. They ease the biofilm proliferation by eating pretty much and grazing on biofilm, and they depurate water in certain cases from the excess of oil, for example. How do we collect samples from groundwater? You can be a caver if you want to be a speleologist and dive down in a cave, but if it's not accessible, you usually use boreholes, which I think in French is piezometer. You have some examples there on the terrestrial side, spiders, pseudoscorpions, and millipedes. Within the aquatic framework, which is more or less the 95% of the focus of my research, you can find cavefish, which are of course blind, copepods, you're familiar with, I'm pretty sure, amphipods, and blind beetles. WA context, I'm pretty sure that you're not super familiar with Western Australia. It's actually, it occupies a huge piece of land. It's a third of Australia's land, and it's the second largest country subdivision in the world. Perth is the capital and hosts 80% of the population. As it's so big, we also have a lot of different climate regimes. North is tropical, south is Mediterranean, and east is arid. I put Paris over there in terms of annual rainfall, and as you can see, is a huge piece of land where there's a much increased aridity compared to temperate Europe. The state hosts 90% of Australian iron ore and 60% of gold reserves, and it's actually the world's largest producer of garnet and zircon. Now, a bit of a guessing game. How many Frances do you think that you can fit in Western Australia? Oh, yeah, very close. 4.5. And the population is Paris intramuros. So you can imagine 4 empty Frances and half of a France with just Paris. Huge piece of land with not many people. Oh, sorry. When you talk about groundwater ecology, because it's so important for the economy of Western Australia, this is what people have in their mind. But of course, it's not all about that. There are also ecosystems, and it's actually probably the biggest biodiversity hotspot in terms of global biodiversity. Just to give you an example, there's 210 calcretes in the Ilgan region, and they host the highest diversity of subterranean beetles in the world. More facts, alluviums in the Pilbara is a global biggest hotspot for stegofauna diversity, and you have there a charismatic blind cave eel, which is actually a cavefish, the Ophisteron candidum. Also, a bit of tributes to the octonauts. They talked about the remipede. You have the only continental alkaline system in the southern hemisphere, and is home of the highly endangered remipede Kumonga XLA. What was our playground for research for the last 6 to 7 years? Star Meadows is a calcrete aquifer. We already know the definition by now, and is actually composed by 110 boreholes, as you met, right? And you have 5 different geological zones over there, playing a nice framework in terms of ecological studies. Is a place that, of course, is in the middle of Western Australia, so climatic regime is quite specific. There's 200 millimeters of rain per year, and the pan evaporation is more than 10 times more than that. So it's very, very arid. So the cyclonic rainfall events in an arid climate regime is actually, if not the most, one of the most studied groundwater ecosystem in the world, with over 30 published papers over the last 15 years. Not exactly down the road. If you think that you have to drive 11 hours by car from Perth to get to there, and which is the easiest way to get there. And this is the road all the way. So very easy to fall asleep. And nice the first time you go there because it's so different, but then it's so boring. Research design. We wanted to check for the influence of rainfall on the ecological dynamics of groundwater ecosystems. So what we did, we framed a site-specific rainfall assessment dividing low rainfall, intermediate rainfall, and high rainfall. And we went in the field 3 times to compare low rainfall and high rainfall. Why? Because the assumption is that in a place which is so arid, rainfall is playing a key role in introducing nutrients within the system that themselves trigger ecological cascades. We used NASA state-of-the-art equipment. As you can see there is a kids fishing rod attached to a plankton net. And since there were so many boreholes, we were able to collect a lot of samples. So the species that you can find there are over there. You have 3 species of subterranean beetles, 3 species of amphipods, copepods, and also oligochaetes and oribatids. This is a video, the only video that I have promised. It lasts 5 seconds, but I just wanted to give you an idea on the biodiversity of the system. So think about Western Australia desert, groundwater ecosystem, and these are 3 samples, which are not the richest samples, it's just the average sample you can find from 3 boreholes. No, it doesn't work. Wait, I probably need to click on it. Okay. So if I told you that this is from a river in southern France, you probably would believe me, right? Okay. So analytical framework. The idea was to unravel the interplay between geology, water, and biota, since groundwater ecology is a small research field but is growing so much. And at the same time, there's so many research questions that still lay. Energy flows, the interaction between microbes, stegofauna, and trophic level assessment. So what we did, we framed an analytical framework that involved genetics, mainly eDNA and eDNA anthropogenic, stable isotope analysis for trophic ecology, and we also used radiocarbon for digging into the carbon flows. This theoretical structure allowed us to have an underground story in 5 acts. We were lucky enough to publish 5 papers, going from microbial patterns all the way to the ecological cascades. I'm going to talk about that a bit during the next 10 slides, let's say. First, we wanted to assess the environmental conditions comparing low rainfall, LR, and high rainfall, HR. What we saw immediately was that there was a change in temperature. Rainfall is usually warmer in this region. In the groundwater, there was a lower pH. Rainfall is usually lower in pH compared to the groundwater that you can find on site. There was also a significant decrease in sulfates due to that pH condition, which were more acidic, and there was also an increase in ammonia and alkalinity. Once we had a look at the DOC, the solid organic carbon, from both a radiocarbon and more of a stable isotope perspective, the framework, the scenario wasn't very exciting. We didn't see many changes, and the carbon load was relatively similar, comparing low rainfall in yellow and high rainfall in blue. But once we had a look at the microbial community through DNA metabarcoding or eDNA approaches, we immediately realized that the microbial community was completely different comparing low rainfall and high rainfall. This was already telling us something. We also carried out some functional ecology studies. We wanted to see which metabolisms were the most prominent under low rainfall versus high rainfall. Again, the scenario wasn't super exciting. As you can see there, the numbers are quite similar in terms of methane metabolism or carbon fixation, for example. But once we had a look at specifically degradative pathways, we found out that during high rainfall, there was a significant increase in metabolic degradative pathways, most of the time for many of them. I would say 7 or 8. So this was telling us that not only the microbial community was different, but there was also a shift in metabolic pathways in the system following a rainfall event. This is just a summary graph to show you the biogeochemical side of things. We realized that there was an input of aromatic compounds, and the microbial community was taking advantage of this input triggered by the rainfall acting as a driver. Next, we had a look at niche dynamics for stegofauna. I will go very briefly onto this, but as you can see already graphically on the right, you can see that the niche hypervolume the stegofauna was occupying was different, comparing low rainfall, the first 2 on the top, and high rainfall, the one at the bottom. We also realized that the soft oxygen was a key driver. So the input of water was also not only transporting nutrients, but there was also an increased dissolved oxygen in a system that is otherwise very stable for many, many days during the year. Then we used stable isotope analysis, both bulk stable isotope analysis, but also carbon-specific, compound-specific stable isotope analysis. And we realized immediately there was a shift in the carbon fingerprint that you can find out from the tissue of all the styrofoam specimens that we collected. 2 trophic levels overall we identified by using conventional nitrogen compound-specific stable isotope analysis, focusing on phenylalanine and glutamic acid. I won't go further in detail. We couldn't work on copepods because if you think that a copepod is probably 0.1 millimeter long in a groundwater, it would have implied that we had to collect probably 5,000 copepods and dry them out and do stable isotope analysis. But realistically, they lay at the bottom of the trophic chain. You do not have photosynthesis, of course, there's no light. Microbes play a key role, and copepods are there on top of microbes. More biogeochemistry. We also did bulk stable isotope analysis. Through Bayesian mixing models, we also found out that, for example, amphipods, if you have a look at the graph bottom left, they base their diet on roots during low rainfall, and there was a very evident shift from roots to particular organic carbon once the rainfall would incorporate within the system. Having a look at top predators, this is a larvae of beetles, of a beetle, actually the biggest species on site. And as you can see, there's a more targeted diet preference towards amphipods once the rainfall is within the system. Talking about the larvae, as you can see, they're bigger than the adults. Apparently not the ideal offspring, or actually yes. In a system where you do not have a lot of nutrients, being big and having those mandibles is actually allowing you to more predatory and more opportunistic behavior, and therefore allowing the offspring to survive. And evolutionary talking, it makes a lot of sense. They're actually called the water tigers, the tizid larvae. Ecological cascades, again, we used a whole set of biogeochemical tools. I won't go further in detail, but we found out that there was a shift in organic carbon incorporations within the diet of the primary consumers, copepods and amphipods. And this was quite important because it was matching, the scenario that we unveiled through microbes. With eDNA metabarcoding approaches, we were able to target the gut content of a copepod, which is again 0.1 millimeter long, and amphipods. And we also found out that there was an increase in alpha protobacteria, which is usually associated with degradative processes. So this story is actually matching what we found in water for the microbial diversity. And very quickly, we also found out that there was a shift in metabolic patterns of the resident microbial community of these primary consumers. This is the final graph summarizing all the tsunami of information that I gave you. Once rainfall incorporates within the system, there's an input of nutrients, which is profited by microbes. Primary consumers, copepods and amphipods, take advantage of this increased microbial community, but themselves are targeted more heavily from by the top predators, which are the beetles. The icing on the cake, as they say in Australia, was to try to find out what was the plant contribution to the diet of the primary consumers, copepods and amphipods. This is important, especially if you consider that once you go as meadows and you collect your samples with your very cheap equipment, what you get is what you see in that picture. You get a lot of roots. So this is a system that has bushland on top, which is often considered as not valuable ecologically. But what we found out through gut content analysis, targeting specifically plant content and plant material, we found out that there was a shift again in their diet, going from more ephemeral plants during low rainfall to more permanent plants. Sorry, to the opposite. During low rainfall, there was a focus on plants like eucalyptus that are more perennial. During high rainfall, there was an input of plant material which was more ephemeral. With this, I conclude the story on the ecological functioning of this system. And just to give you an idea, at this fine scale level, there's not been done a lot on groundwater ecosystems. And this was the novelty of the studies that we carried out. We also worked a lot on eDNA, environmental DNA metabarcoding approaches, and testing eDNA actually for the first time in groundwaters, which this is a publication from probably 1 or 2 years ago. What we found out when we compared morphology versus eDNA approaches, the scenario was quite promising. I think that 14 out of 16 species were detected through DNA. What was interesting, there was of course something missing from eDNA, isotopes and thermosbanaceans, which are the notoriously bad on GenBank for having barcode libraries, barcode references that are not super reliable. From NETH, we were missing soft-body taxa. So what we concluded was that a complementary approach will probably play best in this case and probably overall in molecular ecology. The richest components were the shallow water, where the stegofauna resides, and also the sediment, where you can find a lot of DNA content. Another study was to test the use of passive samplers. You can see bottom left. Again, a very simple setup is just discs that you probably have used for filtering water at some point for biogeochemical analysis. And these discs were left in the water for 10 minutes and 24 hours. We wanted to see if using that passive approach, we could detect the biodiversity on site by not filtering the water actively, but by just dropping those filters. And again, the results were quite promising, indicating that those filters can catch the DNA that is floating in the water and is also matching the morphological assessment. So again, a complementary approach is probably best in these situations. Very quickly, I'll go through hypersaline studies that we carried out over the last 4 years. Hypersaline systems, not sure how much familiar you are. They're quite important from an ecological point of view. Also considering saline lakes account for 44% of inland waters. I actually didn't know about this number, it was quite striking. They provide hugely important ecosystem services. For example, if you think that we all have minerals that are contained in hypersaline waters in our pocket, lithium for batteries, but they're also important for ecotourism if you think about places like Salar de Atacama in Chile. What we wanted to do, we wanted to test eDNA approaches to see if under these conditions that are very challenging for eDNA because you have high UV, high salinity, and also less abundance of biota compared to a freshwater lake. We wanted to see what we could detect by using eDNA approaches in sediment and water, targeting microorganisms, invertebrates, all the way to wading birds. We focused on Rottnest Island. I'm pretty sure you're not familiar with Rottnest Island, but you're probably familiar with quokkas. Have you heard about quokkas? Okay, this is an island that is heavily exploited for tourism because quokkas are very famous and people go there, and there's actually, as soon as you land on the island, there's a map where gives you the selfie spots on the island to take a selfie with the quokka. There was a huge marketing campaign there. Of course, it's not only about quokkas. It's an island that has 5 hypersaline lakes, which are essential for migratory birds coming all the way from Asia. So we wanted to check the microbial diversity on these hypersaline systems, and we did some eDNA studies in water. We found out immediately there was a clustering, very clear clustering across pretty much 3 clusters. And salinity was playing a key role in this, in shaping this cluster. So there was pretty much a lake-driven microbial assemblage where you will have 3 clusters, and the microbial community will go from moderately to extremely halophilic groups. eDNA also helped us in unveiling patterns such as TDS and alkalinity being the main environmental drivers, and through some functional genomic approaches, again, I won't go further in detail because there's no time, increasing salinity meant that were more abundant methanogenomic and aromatic pathways. So overall, these 5 lakes were not one big giant hypersaline lake. They were actually providing heterogeneous environmental framework. We transferred this pretty much the same approach to the Salar de Atacama. Pretty sure you're familiar. We are talking about northern Chile. And we focused also on water and sediment samples. So over there you have a biodiversity wheel, let's say, of the specimens and species that we could find. We applied a multi-assay design. We targeted invertebrates, vertebrates, and wading birds. And we were able to detect a relatively big range of resident hypersaline taxa, including rotifers, endangered snails or amphipods, and also flamingos. So eDNA can also provide information on the use of habitat by wading birds. Next step, quickly, we wanted to compare how good was the eDNA assessment compared to the classic conventional morphological assessment. And this table is summarizing the main outcomes. So compared to net-based assessment, eDNA allowed a more refined taxonomic assessment for example, copepods, ostracods. But at the same time, it's not a perfect tool. There were certain taxa such as Ephidridae or Iridinae that were not detected at all through molecular tests. As a result, a concept that I already stressed, eDNA is really powerful and a complementary tool. Very quickly, provocatively, I named this slide Escaping from Gene Bank. For groundwater ecology studies, if you think that just 15 to 25% of the scarce subterranean species are represented, that's why pretty much we're escaping from Gene Bank. But how are we doing this? We are trying to design barcode reference libraries. So with the specimens that we have the specific barcode referring to that specific vulture specimen, often from the museum. And what we're trying to assemble is also, in order to elevate even beyond barcode reference library, we're trying to assemble the first stegofaunal mitogenome database in order to have also a vast set of phylogenetic information there. We are also exploring new approaches. We will use biosensors to detect cave fish. We will target PFAS in groundwater biota. This is a hugely understudied topic. And as we all know, PFAS are everywhere. And groundwater is providing a huge chunk of the water we use. And in collaboration, especially with Tariq, we are going to, through a PhD student, we are going to apply for the first time fatty acid analysis in groundwater biota. And eRNA. Are you familiar with eRNA? Have you heard about eRNA? Environmental RNA is the concept of DNA applied to RNA. eDNA targets non-expressed sequences. eRNA focuses on the expressed metabolisms. Very difficult. There's already papers saying, is it worth the extra mile? Because they found out that eDNA was actually comparable in terms of results to the eDNA, but we will dive into that. Very quickly, some projects I'm currently involved with, eDNA, which is the first global eDNA study on lakes involving 500 collaborators, a few biodiversity projects on groundwater ecologies, Stegofauna Mundi, which is the biggest Stegofauna data set to date, and plastics, which is the first microplastic study, global microplastic study in groundwaters, and 3 more specific initiatives that are more related to BOREA. I'm the leader of this initiative on delivering the first eDNA global study on groundwaters called GREG. There'll be a seminar on the 25th of June, online seminar. If you're interested, let me know. We are in the process of establishing a protocol d'accord between Curtin and the museum in order to ease interaction and foster interchange of knowledge. And there's also through the CNRS, not probably this round, but also the next round, there's a set of actions that I am to apply to, and also a joint grant application with Nicolas on freshwater ecology. Just to conclude, this is a quote from a recent paper on eDNA to 2014, if you think that eDNA exploded really from 2012. This is exactly what they wrote in the paper. And as you can see, you can read through it, and as you can read, there's a very enthusiastic and positive feeling behind it. This is the enthusiasm of finding a new tool. Of course, reality is different, but at the same time, I personally completely acknowledge that eDNA is a very powerful tool. It's not the best tool on earth, but it's definitely an arrow in our quiver that we can use to improve ecological assessment, same as stable isotopes. Because at the end of the day, and here I quote a recent perspective paper that was published probably 2 weeks ago, ecosystem ecology, I strongly believe, needs also an ecology of molecules. With this, I conclude. I thank you, and sorry if I was too late. 43 minutes, is it okay? It's fine. Perfect. More than keen on collaboration, questions, doubts, and sharing of expertise. And thank you for having me again.
Thank you very much.